Hi all, welcome back. And now we are going to continue talking about cells. And now we're going to talk about, though, we already talked about the history of cells, prokaryotic cells, eukaryotic cells, and their organelles. Now we're going to talk about cell function. Like, how do cells actually work? How does material get into the cell? How does it exit the cell? What's kind of the functionality around the cell? And then do different cells have kind of different functions and what makes them kind of work? All right, so passage into and out of the cell. Okay, for some things, that cell membrane. Okay, we talked about it before, that phosphate head, that fatty acid tail. It allows some things to move across kind of freely. Mainly things that don't carry a charge can move freely. And then there are special proteins that occur in the cell membrane that'll allow things to just move freely across. Like, for example, water can move freely across the cell membrane because there's a protein in the cell membrane called an aquaporin. The aquaporin allows for water to just move back and forth through a process which we call osmosis. And so that's just the movement of water across the membrane. Okay? But for things that might have a charge, which is a lot of things, or things that might be large and can't fit through simple tiny protein channels, um, we need to talk about how they get in. And so that's what we're going to talk about now is kind of bulk substances and how they move in. Okay. So two kind of broad terms. One called endocytosis. Okay, endocytosis means that you're engulfing, or the cell is engulfing a substance. Okay, from outside and bringing it in, and it's becomes part or brought into the cell in a membrane. Okay, so it's like the membrane of the cell opens up, attaches, and brings it in, and part of that membrane, now it's in this membrane capsule or vesicle. Okay, exocytosis means that you're taking something from inside, typically bound in a membrane, and it comes out and then gets spit out of the cell or leaves the cell, and the membrane becomes part of the cell membrane. Okay, and so I'll show you what I'm talking about here in a second. Okay, so two other processes that can occur, one we call autophagy, okay, which means that you're destroying self-made material. Okay, and so something like this, I kind of already talked about this last lecture. Let's say you have a damaged mitochondria, so it's not working very well. Your lysosomes then can attach to it. The membranes will kind of attach. The lysosome will push digestive enzymes. Those digestive enzymes will destroy the proteins and the lipids and different things. And then the components can be recycled to build a new mitochondria to replace the old mitochondria. Okay? Phagocytosis. Okay? This is that one of those endocytosis, meaning bringing it in. Okay, so you can get this material, maybe it's foreign material, and this is a white blood cell and it's destroying it. Maybe it's a food particle, something like that. But the membrane will wrap around the material. It'll bring it in, and then something like a lysosome can then attach to it, destroy it, or break it down into smaller parts. And then those parts can be used as either energy or used to build new organelles or the function of the cell. Okay. So, endocytosis, again, phagocytosis. A couple things with phagocytosis. Phagocytosis is what we refer to a type of endocytosis that we're dealing with solid matter. Okay, so solid matter, maybe this is a bacteria, this is a white blood cell. okay, that white blood cell is going to consume that bacteria, then it will destroy it, okay? But it's a solid matter, okay? Pinocytosis means it's liquid matter, okay? So maybe this is some type of, you know, liquid-based material, okay? So maybe it's something that's dissolved in kind of water. Maybe it's a carbohydrate, a sugar water, or something like that. Um, it it would then engulf those liquid droplets, okay, and bring it in as part of the system, okay? So phagocytosis, solid matter; pinocytosis, liquid matter. Exocytosis is just kind of what it sounds like, exit. Okay, so you want to get rid of it if it's waste material, or sometimes maybe not waste, but maybe it's product that cell's made. Maybe that cell's made a bunch of new proteins, or maybe that cell's made a bunch of lipids like hormones, and that cell is just cranking out these hormones, but it produces them in these secretory vesicles, and then those vesicles, like I said before, the membranes will bind together. Okay, and when the membrane binds together, the material can leave and that vesicle just becomes part of the membrane. This is the great kind of component with that fluid mosaic, is that it has the ability to stretch. So you can add new phospholipids to it. You can take phospholipids away. So if you're going to engulf something, bring it in, you're going to take phospholipids away, the cell's going to shrink a little bit. Okay, if you're going to expel material, the cell's going to expand a little bit. Okay, and so here you can see the releasing. So this vesicle here, secretory vesicle, is releasing this material, and the membranes have now merged together and are part of one now. Now, not all the time is it so free flowing kind of thing, like things getting engulfed, brought in, things getting expelled. Okay, often you only want certain things coming in or only want certain things going out. And so what happens is what we call receptor mediated endocytosis or receptor mediated exocytosis. Okay? Which means that you have protein receptors that occur kind of in a little pit, and when the target material is there, maybe this is a liquid, maybe this is a hormone, maybe this is small proteins, when they get associated with this receptor protein, that can cause a shape conformation change. And so it can cause the pit to increase in size, and then it will pinch off. And then you have this coated vesicle that can come in. You can get the opposite happening on the way out. Okay, you'd have proteins that are facing the opposite direction, receptor proteins, and material that's inside form it, and then that pit forms the other way, bubbles out, and releases. Okay, and this is just an electro graph or electro microscope picture of one of those functioning. Okay, so you get material that's forming and attaching to the protein receptors. Okay, that causes the shape to change. Then it just folds over on itself, pinches off, and then now that material's inside the cell. Okay, a lot of that so far is what we call passive transport. Okay, passive transport just means that it occurs without energy. Okay? Active transport, on the other hand, means that you have to supply, in most cases, ATP in order for that to occur. Okay? This can happen with those protein channels that we talked about before, where you have these proteins that are embedded in the cell membrane, and they're allowing different things to go in and out of the cell. Okay, but sometimes the only way that protein channel is going to open up is if you use energy to supply that channel. You supply that channel with energy, and that opens the port, changes the shape. Okay, and this is how a lot of pumps work. Okay, so the energy is used to drive the pump, and so it can pump substances against concentration gradients. Okay, so most substances move from high concentration to low. Okay, so if you have a bunch of sodium on the outside, it wants to move to the inside. But what if we wanted to move more sodium out? Okay, you have to supply energy to do that. Okay, and that's where active transports come in. Okay, so it really allows for you to get even higher concentrations in certain regions. And we'll talk about this when we talk about how action potentials work, how nerves work, okay? And we'll go into much greater detail, but that's part of that active transport, okay? And one such active transport is exactly that, how muscles and nerves work, the sodium potassium pump. Right? And so it, again, is an active transport. So real quick, we'll just cover it briefly on kind of how this works, and then we'll dive in to how action potentials and other things are formed later. Okay? So sodium and potassium pump. It uses energy from ATP. Remember we talked about this before. ATP is generated in the mitochondria. Okay? And or in prokaryotes can have sodium potassium pumps also, and ATP would just be generated in the cytoplasm. But we're talking mainly about humans. Ours is generated for the most part in the mitochondria, a little bit in the cytoplasm, but not much, in the mitochondria, and that energy is used to pump 3 sodium out of the cell and pump 2 potassium into the cell. Okay. When we look at this, about a third of your energy, okay, so if you're looking at a diet of maybe, let's see, 2,100 calories, somewhere in there, about 700 calories will be used to just do this process right here, this pump. So a third of the energy your body takes in is used to drive sodium potassium pumps. Okay, so here it is, ATP. It's going to come up, it's going to have a binding site. One of the phosphates is used. Okay, so ATP stands for adenosine triphosphate, 3 phosphates. Okay, one of them is used, it's broken off, the energy is used from it. Okay, so now you have adenosine diphosphate, 2 phosphates, and in an inorganic phosphate group that's left, okay, after this process. But that energy allows for this channel to push sodium, sodium, okay, from inside the cell out of the cell. Okay, so this is inside. Okay, so sodium's pushed out of the cell, and potassium is pushed into the cell. Okay, now there's way more sodium out here, okay, than there is in here, and vice versa. There's a lot more potassium in here than there is out here. So these are pushed against the gradients. Okay? So sodium is being pushed from low concentration to high, and potassium from low to high. That's why we need ATP. If it's the opposite direction, it just flows typically from high to low. Okay? We'll talk more about this. Okay? This is why individuals who say you have cramps, leg cramps, athletes, and people say, well, you need to eat a banana, you're low on potassium. Okay? Really, you could be low on sodium, but not very many people are. Okay? Potassium is a little more limited in our diet, so they're probably right. If you're getting cramps, it's because you don't have enough potassium for this channel to work. So your muscle, instead of it firing and then relaxing, it fires and then it stays. Okay, and you get what we often call Charlie horses or cramps. Okay, and it stays flexed. Okay, now it could be sodium is lacking, but most of the time, again, it's potassium because there's less potassium in our normal diet than sodium. Most most of us have plenty of sodium in our diet. That this is not the limiting resource that potassium would be. But again, we'll come back and talk about these later. All right, so cell shapes. Okay, there are about 200 different types of cells in the human body, and they all vary in shape and size and function. Okay, some of them we're going to talk about, and it'll be important for you to know them because the shape or kind of the structure will determine its function, like we talked about a little bit before. Often, shape or structure determines function, okay? But cells combine together, right? Exchanging information, exchanging product, waste, that kind of stuff, is what makes tissues. And then tissues, multiple tissues, and many times interacting with each other is what makes organs. Okay? And so there are lots of cells that we will talk about in coming up lectures that are kind of important. So whether that be like the shape or size of the structure, like squamous or thin, flat, scaly cells, cubu cuboidal or squarish looking cells, columnar or long, kind of wide column-like cells, okay? Polygonal, stellate, ovoid, or spheroid, okay? All kinds of different shapes of cells. Sometimes the function is perfectly fit for that cell. Other times, the cell might change shape over time. Okay? Discoidal, fusiform, fibrous. Okay? All of these different cells will have different. They obviously have different shapes because they have different names for the shape, but they also have a lot of times they'll have different functions. Okay? And the function of the cell, it needs to be fibrous. Okay? Your muscle cells need to be fibrous. They need to be long, thread-like cells in order for them to relax and contract. Okay, and so relax, contract. And so you need these thread-like fibrous cells. Okay. All right. Let's look at what some of these kind of look like. Squamosal. Okay, so kind of flat, scaly-like. You might think of these as skin cells. Cuboidal. They look like cubes. Okay. Columnar. Okay, so they look like cubes, a little bit more elongated. Often things like your intestinal tract will have these with microvilli or little projections that are hanging off of them. Okay, polygonal, stellate or star-shaped. Okay, nerves we get labeled nerve cells, kind of stellate, spheroidal or spheres. Okay, a lot of your white blood cells might be similar to this. Okay, discoidal. Okay, your red blood cells have kind of an indentation. Okay, fusiform, fibrosa. Okay, again, we'll talk about a lot of these as we go, but that's where the names are really coming from. And so often you'll see a name associated with a cell type, and you can come back to kind of this um, lecture and or revisit this lecture to find out, oh, okay, that's what we're talking about, that kind of cell, that kind of shape. Okay, and it just helps with visualizing where those cells would be, what kind of their function is as we progress. All right, just general information real quick about human cells. Okay, so most human cells are about 10 to 15 micrometers in diameter. Okay, so 1 micrometer is 1 thousandths of a millimeter. And so if you've ever looked at like a ruler, and you see on one side, it's lots of rulers that we hand out to students today. They have one side that's inches, okay, and then the other side is centimeters. But each one of those little teeny kind of marks, okay, so on your way up to 1 centimeter, you're going to have 10 little teeny dash marks, okay. Each one of those is 1 millimeter. Okay, and so a micrometer is 1,000th of that millimeter. Okay, they're pretty small. Okay, so 1 micron or micrometer is 1,000th of a millimeter. Okay, egg cells, which are some of the biggest cells that humans have in their body, are about 100 micrometers in diameter. Okay, some nerve cells are even much longer, much bigger. Okay, they can be a meter long. Okay, so remember, that's that surface to volume ratio. There's a lot of surface area, and they're really thin, and that really allows them to be extend very long. So 1 meter, roughly 3 feet long or so. For those of you who still are working with feet and inches. Okay, so fairly long structures. Yeah. And of course, like we talked about before, there's a limit to the cell size. Okay, and it's totally based on that surface to volume ratio. Again, we just kind of talked about this. Most cells are not making all the energy that they need in order to survive. They have to get their energy, they have to get things from outside the cell. Okay, and that's limited by how many proteins you can stick in that cell membrane, in that surface area. Okay, if you make a bunch of folds, then you can put more proteins and you can get more things in. Okay? But if you're just a spherical cell, then you're limited on how much material you can come in. Okay? And so one of that key components is large cells really can't support themselves. Okay? Not only that, is when that volume gets really large, then you get that material, anytime it flexes more, and you can start to get holes or ruptures that form inside. Okay, okay, so just to recap, we already talked about this a bunch of times. Okay, and it's just a really important concept. There's a lot of students that believe elephants have bigger cells than humans, and humans have bigger cells than flies, and flies have bigger cells than, you know, uh, I don't know, like a housefly has a bigger cells than fruit fly or something like that. But that's not true. They just have more cells. Not size-wise, cells are pretty uniform. They're pretty close to kind of this size. Volume, again, has to do with that surface area. Um, and so that really comes in. The volume is proportional to the cube of the diameter. The surface area is proportional to the square of the diameter. Okay, so volume increases faster than surface area when the cell increases in size. Okay, next time we'll talk a little more about tissues. So how do these cells link together to form tissues? And then how do those tissues form organs? Next time.